Hi you guys, we're starting blood flow 1 still, but this is video number 3. And we're switching now to arteries. Let's think about some of the functions of arteries. Look at this diagram here. We have an artery that is moving 4 liters of blood per minute, and it's branching out to 4 smaller arterioles, and each one of them is transporting about 1 liter per minute. That makes sense, right? So this blood flow is equal between them. And if you think of an organ that is sort of downstream from these arterioles, they're all receiving the same amount of blood. So that can happen. Or we could have a change in B, for example. And here is vasoconstriction. So this is narrowed. And then blood isn't able to enter at the same speed here, and it will back up and go out through A or C or D. So the change here in B, it's dropped down to about 1/4 liter per minute, whereas the other ones have increased by 1¼ liter per minute in each one of them, still adding up to a total of 4, but we've redistributed that blood. So imagine you're out doing exercise. Imagine that right here is, downstream from B is your digestive system, and this is maybe going to muscles. So you're able to successfully redistribute blood given the particular homeostasis that you need to be in. You need more oxygen coming to your muscle at that particular time. So it's interesting to think about how blood distributes ordinarily and how it does it at rest and during exercise. These are not numbers you're going to memorize, they're just sort of fun to look at. You take tissue like the heart, for example, and let's backtrack here, the heart, where 4% of the blood would be within the heart at rest. And it goes up during exercise to about 4.4%, which isn't huge. It's not a very big change because it can't pump more than that volume anyways. The GI tract, however, at rest is receiving about 27% of that blood, and now you go out and exercise and that's going to drop down to about 4.8, and then you're redistributing that blood is going to muscles instead. So that's a big change. Let's look at some others. The kidneys. Kidneys are used to filtering your blood, and they continue to filter. They're receiving 20% of that blood, but now again, you're out exercising. You're going to need that oxygen in the muscle. You're going to need that blood elsewhere, and it drops down to about 4.4, which is similar to the heart. Skeletal muscle. Look at this jump from 21% at rest to 64%. Get that oxygen and make ATP. The skin goes from about 5 to 13 or 14, and this is because you are bringing blood up to the surface in order to lose heat. So it's a form of losing heat. And bone and other goes from 9 to 4. I don't have a whole lot to say about that one. This is a reminder, what kind of capillary is this? Continuous, fenestrated, sinusoidal, or messed up? And the answer is sinusoidal. Let's go to veins now. Learn a little bit about veins. The majority of the blood is in veins, about 65%, and a very important anatomical part of veins is that they have valves, and valves prevent backflow. Very important. Imagine if we didn't have valves, we would probably have blood pooling in our limbs, in our extremities, in our ankles, because blood in a vein is going to be at a lower pressure, and it has to flow against gravity back up to the heart. So it is a difficult feat. So what it does is veins are going to depend on skeletal muscle to be able to squeeze and pump that blood past those valves and eventually make it up to the heart. This is a really good picture of that. Get my pen back. And in this case here, you see how the vein travels between large muscle groups. and you have a valve here, and you have another valve here. So when this muscle contracts, it can push this blood beyond it, and then blood's not going to continue to trickle back down to the ankle. Here's that diagram. You've contracted that muscle, you squeeze that vein just because it's in that location. It opens these valves and blood will shoot up beyond that valve. And as it does that, it continues to move up and up beyond the new sets of valves that are beyond this piece here. Very important. So sometimes valves don't work well. So a pathology of valves are varicose veins. This is an example of varicose veins where the blood is pooling, and it isn't just unsightly, but it can be painful. So one possibility is to actually remove them. But then where does the blood go? Well, it goes to the neighboring veins, and hopefully there isn't weakness in those valves too, allowing blood to pump back up into the heart. Let's talk a little bit about angiogenesis. So angio is vessel, genesis is originate. So angiogenesis is the creation of new blood vessels. When do you need these? Well, blood vessels are going to grow when you grow. If you have an injury, they need to be replaced or repaired. Another example is in the uterine lining after menstruation. These need to be new networks of vessels need to be manufactured essentially. And during endurance training, remember we talked about this, is how do you get your VO2 max higher? You get it higher because you're able to move more oxygen. And one way of doing that is to increase your vascularization. So create angiogenesis. Another example is cancer. Imagine a group of cells and this tumor is getting larger, and you need to irrigate that tumor if those cells are to stay alive. So they create their own vessels. How is this regulated? So it's controlled by chemicals, by cytokines, which are local hormones. And they're called growth factors or mitogens when they induce mitosis. So you want to create more of them. You're exercising a lot. You need angiogenesis in these muscles. And these chemicals then are called mitogens. They're called statins when it's a chemical that actually prevents mitosis of these vessels. In other words, you're not making new vessels. So there's a control in the body of yes, make vessels, or no, don't make vessels. What's the applicability of this? This is very interesting because if we could have these endostatins, if we could have this chemical that stops the growth, it stops angiogenesis, then can we treat a cancer? Because if we were to remove the ability of a tumor to create its own irrigation, then we would essentially strangle the tumor. And also, what about mitogens? If we could control that also, could we treat coronary artery disease? I mean, maybe we need new coronary arteries that are not clogged. And so if we could play with mitogens and statins here in the angiogenesis, we could solve some serious pathologies. There is a little bit of research here done on cutting off a tumor supply in the war against cancer, and I wanted to show you that graph. So this is an endostatin target dividing blood vessel cells. So we have an endostatin delivered, and this tumor growth, it's going to grow, and then the endostatin sort of kicks in and it drops. And then it starts to grow again, endostatin gets treatment again, it grows, it drops. And so on and so forth for these treatments. And after about 4 of these, this was done in mice, that tumor failed to grow. The tumor volume stayed low because there was no angiogenesis happening. So this seemed to be very, sort of show a lot of promise in the research, in cancer research. I want to compare this graph with the one down below here, where you had traditional chemotherapy drugs, and you had the untreated patient had this huge growth of this tumor, and the treated one would seemingly look good in the beginning, but after a while, even with these treatments that were going on of traditional chemotherapy, that would rise again. So as I was saying, these treatments here showed a lot of promise in mice, and they have had varying results in humans. So some part of it isn't well figured out yet, but they're still working on this. What about if we could stimulate angiogenesis during heart disease? So imagine here on the left a heart that has atherosclerosis. This is cholesterol in the artery, and it's clogged to the point that no blood is arriving here to this section. If we could actually stimulate angiogenesis in these vessels here, look what happens in B. In B, this has been stimulated, and they've been able to reach out to the area that was once clogged. So that continues to be clogged. But with the growth of the collateral ones, you could actually irrigate that area. So these are really interesting research possibilities with angiogenesis. And that's where we stop. It's the end of blood flow 1. So we have 3 videos for this particular section. Thanks for listening, guys. Bye.